Hello all, welcome back. We're about to start our cancer early detection session. My name is Cristina Zaveleta and I'm an assistant professor in the biomedical engineering department at the University of Southern California. My co-moderator is Jesse Jokerst, and he's an associate professor in the Department of Nanoengineering at the University of California in San Diego. Jesse and I spent our postdoctoral fellowship together in Sam's lab, and we're both really excited to be reunited here to moderate this incredible session on cancer early detection. Sam's greatest ambition was to change the nature of cancer diagnosis. He would often speak of cancer progression as a movie, where today we are only able to look at a few scattered frames right when the movie is about to end. He spent his career focused on the vision of being able to capture the whole cancer movie, including footage of the very first rogue cells. It was that vision that led him to partner with Don Listwin to found the Canary Cancer Center for Early Detection at Stanford, which aimed to bring together an interdisciplinary mix of diverse researchers who carry on Sam's vision by generating groundbreaking research with the mission to utilize diagnostic strategies for the detection of cancers at early curable stages. In this session, we will be hearing from some of our thought leaders in the field of cancer research who share in Sam's vision and continue to promote the early detection of cancer. It's my pleasure to start off our session by introducing you to the director of the National Cancer Institute, Dr. Ned Sharpless. Dr. Sharpless was officially sworn in as the 15th director of the National Cancer Institute on October 17, 2017. Prior to his appointment, he served as the director of the Lineberger Comprehensive Cancer Center at the University of North Carolina. He has authored more than 160 original scientific papers, reviews, and book chapters. He's an inventor on 10 patents, co-founded 2 clinical stage biotech companies. And Dr. Sharpless is a member of the Association of American Physicians and the American Society for Clinical Investigation, and a fellow of the Academy of the American Association for Cancer Research. With that, I am delighted to introduce our first speaker for our cancer early detection session, Dr. Ned Sharpless. Thank you for joining.
Great. Thank you for having me. And I guess good morning out there. Good afternoon to me. It's a real pleasure to be here. I wish it were in person. I'd love to be out in Palo Alto again. I'm very excited to be part of this really important panel with such outstanding speakers and talking about this very important topic of cancer early detection, which is certainly one of the areas of Sam's life's work and an area where his science and clever ideas really made a difference. And also, I think, an area where there's tremendous opportunity in cancer research right now. This is something the National Cancer Institute thinks about deeply. We really believe that if we want to get to President Biden's goal, and I'm sure many of you have heard the president say this, that he would like to end cancer as we know it, then cancer detection and cancer prevention are certainly big parts of that goal, and would play a key role in any of that sort of real population-wide progress against cancer mortality that we all really so strongly seek. So I think this is a great way to commemorate a fabulous scientist talking about such an important topic of such incredible value to the public health. I'm sure many will talk about Sam's great science and the great ideas he had and his clever biology. And I think we've also had some focus on his really extraordinary record as a mentor and training a real generation of scientists and engineers who have taken on these approaches to cancer research. I thought I would emphasize perhaps something else that's slightly different to start with, and that's Sam's interaction with early commercialization of science. Next slide, please. So Sam was a real exponent of, it wasn't enough just to have a great idea and publish a great paper, but you really had to translate those wonderful ideas into benefit for the patient. And so that was really this year, deploying cutting edge technologies to the problem of cancer early detection. As noted here, Sam was involved in several early stage companies that try to commercialize early detection technologies. And as you know, in particular, Sam was involved in the founding of CellSite Technologies, and CellSite was an awardee of the NCI's Small Business Innovation Research Program, and that's a grants program they have to help small companies commercialize ideas to achieve commercial success in the name of reducing the impact of cancer on people's lives. With SBIR support, CellSite is developing an imaging technology that helps clinicians determine patient response to immunotherapy during early stage treatment. And Aruna Gambhir, Sam's wife and the company's CEO, who spoke earlier in the symposium, has really leveraged SBI support to take a product from a concept to clinical trials. And Aruna has been a real champion of the SBI program and a friend of the NCI, and has encouraged women in entrepreneurship. And we really tremendously value her support of this program. SBR program is about 3% of the NCI's external budget. It's a really important thing that we do that I don't really think, frankly, gets enough credit. It's a very strong program. Next slide. It's two sort of linked programs, the SBR program and the small business technology transfer programs, and they're highly competitive programs that encourage domestic small businesses to engage in federal research and development with the potential for commercialization. And its support differs from that of the private sector. So private sector investors tend to invest in businesses with an established track record, and the NCI, on the other hand, is really can take on out-of-the-box ideas and fund innovative stuff and early proof of concept stage stuff and ideas that might not have a clear commercial path. I think we've had a lot of success with very innovative device companies, for example, that have trouble commercializing their ideas as opposed to straight up therapeutics. And these funds can really be essential for bringing these projects to patient benefit in a way that is non-dilutive to the company. And as you can see from this slide, there are several important benefits for companies that receive an SBR award. And CellSite is one of many NCR SBR companies that have been very successful. And these include things like Illumina, Seattle Genetics, G1 Therapeutics, a company I founded. Most recently, a company called Immunomedics, which was acquired by Gilead for $21 million for their recently FDA-approved molecule, sacituzumab, for triple-negative breast cancer. We've done a recent analysis of the SBR program using sort of standard venture capital metrics of productivity, and the return on investment, we believe, is on the order of 33-fold. It's quite strong in terms of its leading to venture-backed and publicly backed companies. And so I think it's a real success story that we don't talk about much in the NCI. And I think it was something that Sam used to real avail. I'll say one more word about it before I turn to science. Next slide, please. And that is, we reconvened a group of experts to give us advice on the SBR program. And one of their top recommendations was this new concept award, for which we have just put out a solicitation for proposals. I know there are many at Stanford with innovative ideas. And if you're thinking about starting a company, this might be an award for you. It's a contract award to encourage small businesses to work on bold and outside of the box ideas to make significant impact on the prevention, diagnosis, treatment, and care of pediatric or rare cancers. And it can provide $300,000 in funding to small businesses to explore feasibility and demonstrate proof of concept. And there's more information on this slide. And I encourage those of you who are interested to look into it. Next slide. Now, let me talk a little bit about the work at the NCI around cancer prevention and early diagnosis. And this year, I think, marks the 50th anniversary of the National Cancer Act. So President Nixon, in this grainy photo from 1971, is signing this law that didn't create the National Cancer Institute, but it gave the NCI a bunch of new capabilities for cancer research that created the modern NCI that we know, that includes cancer centers and the SEER database, and revamped the clinical trials network, and it created Frederick National Lab, and it did lots and lots of other things that really made modern cancer research what it is in the United States. Perhaps most important was the Cancer Act made cancer diagnosis that we could talk about publicly. So prior to 1971, this was a shameful diagnosis. There was stigma associated with the cancer diagnosis. And since this act and the forthright statements of people like Mary Lasker, who helped support this bill, cancer is now a disease that we can talk about and we can make progress on. I think it's also important to say that in some ways we have not succeeded the goals of the National Cancer Act. So President Nixon was told that we'd probably have a cure for cancer, say by 1976 for the bicentennial anniversary of the country. Obviously, cancer turned out to be a much more difficult problem than that. But now, 50 years later, I think it is a good time to take stock of the progress we've made in cancer research, and certainly the opportunities and challenges that still face us. Next slide. One important metric that we follow very carefully at the National Cancer Institute in terms of cancer data are shown here in the annual report to the nation. This is a document the NCI, the CDC, the American Cancer Society, and NACER, which is the National Association of Cancer Registries, put together every year. And I think of it as like the scorecard of how we're doing against cancer. And we look at both incidence and mortality. I'm showing the data for our most recent cancer mortality statistics. And you can see that in many ways, the news in cancer research is good. We've seen a decline in cancer mortality, some really marked declines in certain kinds of cancer like melanoma and lung cancer shown in the blue bars in the bottom. But you also see here that there are some areas that need additional progress. So you see cancers like pancreatic cancer, where there's not much change in the mortality, and there aren't a lot of new therapeutic ideas, and certainly an area where we need more work. And you even see some cancers that predominantly are obesity-associated cancers that are increasing in mortality in the United States, things like endometrial cancer. And so certainly an important thing for the NCI to watch as well. Cancer mortality in the United States has been declining since the 1990s, but that rate of decline has sharply, I would say, increased in the last few years. And I think this reflects both generations of tobacco control and other cancer prevention measures, plus successful cancer screening, things like colonoscopy and pap smears and mammograms, plus now a clear effective therapy. So the lung cancer mortality numbers, the lung cancer and melanoma numbers are so good because of effective therapeutics in those diseases. So immunotherapy and kinase inhibitors and stuff like that. So really it's this, all these different approaches converging to make progress against cancer that are starting to benefit the public health. Next slide. I'll mention a few scientific initiatives that I think are relevant to today's discussion. One is the RAS Initiative. A primary goal of this initiative, based in Frederick National Lab, is to develop inhibitors of RAS, but also RAS signaling pathway, but also to provide a lot of reagents and tools for the community that wants to work on RAS signaling. And this is something that we have done in great detail, including most recently delivering on a real priority of the RAS Initiative is 256 validated targeted mass spec multiplexed assays, many with antibodies for quantifying protein expression and phosphorylation and activation in all the various RAS downstream signaling networks. These assays replace over 60 Western blots with measurement of high molecular specificity and quantitative precision, and are now widely available to the community with fit-for-purpose SOPs that can be found at assays.cancer.gov or antibodies.cancer.gov for those of you who are interested in these reagents. There's also on our proteomics assay portal more than 2,500 assays available. This portal now has more than 7,000 users a month, listing all these monoclonal antibodies the NCI provides for this kind of research. So it's sort of how we provide infrastructure to make cancer science possible. Next slide. One of an important proteogenomic example is from CPTAC. This is our proteogenomics consortium that is studying the genomics and RNA expression status and protein status of cancers. CPTAC has had a marvelous run now with high-profile publications on something like more than 14 different papers recently, including this recent paper on adult glioblastoma, identifying new signaling networks within glioma and new subtypes of glioma, particularly with regard to immune profiling of the microenvironment of these tumors. As many of you, I think you are aware, the immunotherapy track record in glioma is quite mixed and without terrific evidence of response in most glioma subtypes. And so I think this appreciation of immunologic heterogeneity among glioma subtypes could be very important. This is one of many CPTAC papers. We recently had a similar paper on pediatric brain tumors, and these data now are all available through the NCI proteogenomics portal, and are it's great to have these matched data sets of RNA, DNA, protein, highly multiplex protein assays, et cetera. So again, additional support for the research community. Next slide. Let me talk a little bit more about specific NCI efforts on cancer screening. So this is data from the PROSPER Consortium, which is a population-based research to optimize the screening process. PROSPER collects data on something like about 11 million people in the United States. So these are data from like 1 in every 30 Americans. And these are the kind of data we have in the PROSPER consortia, which we have for breast cancer screening, lung cancer screening, cervical cancer screening, and lung cancer screening. And here's evidence of the effect of the pandemic on cancer screening, and you can see it's quite dramatic. So shown on the left is the effect on mammography, with that almost going to zero nationally. So we saw a more than 95% decline in elective screening for breast cancer in the early stages of the pandemic. That has largely recovered now. We see a similar effect with colon cancer screening, shown on the right here, with colonoscopy sharply declining. That also has largely recovered in the United States. But in both instances, we think that we missed on the order of 10 million screening events in the United States during the pandemic in 20 and 21. And the capacity to make that up doesn't really exist in the United States. So we just are resigning ourselves to the fact that we are going to see cancer being diagnosed at later stage and so-called upstaging. And I think as a community, we have to take collective action to try and make the effect of the pandemic as least bad for our patients as possible. by making sure that we make up screening where appropriate and get those patients with early stage diagnoses in for treatment as quickly as possible. Many of the societies interested in cancer, such as ASCO and the American College of Surgeons, have put out guidelines and a variety of pandemic-related materials for the care of cancer patients or new cancer diagnoses. And I think that really as a collective group, we need to take action to make sure things are as not bad for our patients as possible. Also, there is an interesting scientific data finding here, which is if you look at the colon data on the right, you see that orange line that is not nearly as badly affected as some of the other modalities. And that's FIT testing, that is fecal immunohistochemical testing, which is home-based screening for colorectal cancer. The NCI has long been interested in the idea of home-based screening for cancer in general, in cervical cancer and colon cancer and other kinds of cancer. And so this is an important advantage, if you will, of home-based screening, and another reason why we think we need to move some kinds of screening from clinic-based screening like mammography and lung cancer screening. We will try in some instances to try and move this to home-based screening, which could also be of tremendous benefit outside of the United States, for example, in low and middle income countries where screening is not so as prevalent. Next slide. I'd also say a little more work about the work the NCI has done related to the pandemic. We had received funding from Congress to work on a serologic research. So the $306 million of a special appropriation. So in no case did we use cancer research monies to study COVID, but with this extra funding from Congress, we did a number of things to try and really understand the immune response to coronavirus infection. I think it's probably obvious to most of you that the NCI has a long and distinguished history of virology research. So, Bob Gallo, Harold Varmus, et cetera, and many really important discoveries like the discovery of HIV, causative agent of AIDS, and the first effective anti-HIV therapeutic. And importantly, Doug Lowy and John Schiller invented the HPV vaccine. And so the NCI has a lot of capabilities for serologic research. And so it wasn't a big deal for us to pivot our serology capabilities to COVID work. And we still now have a bunch of ongoing lively things, including profiling assays for the FDA. So in a sort of first ever intergovernmental consortia, we did proficiency testing for the FDA on these, many hundreds of antibody assays that were being developed by manufacturers and sort of unleashed on the American market, and showed that some of these tests were quite useful and some were not so much. And so I think this has been an important example of how Frederick National Lab and the National Cancer Center, National Cancer Institute rather, can sort of help with this kind of research when needed. This effort has been described by Jeff Shuren, the leader of the Device Center at the FDA, in a recent review in the New England Journal that is of interest if you're interested in sort of how a government can work together on proficiency testing. And I think we may be doing more of this in the future, given the success of this effort. Next slide, please. I'd like to mention, the kind of impact the NCI can have on national lung cancer screening guidelines. So I think many of you are aware of the National Lung Cancer Screening Trial, which was an NCI-led trial to study the ability of helical CT scanning to detect chest CT to detect early lung cancer. And this was a positive trial, and a similar result was obtained by a trial in Europe. And now we have recommended lung cancer screening for certain individuals, people of a certain age who have a certain history of tobacco use, as a way to reduce lung cancer mortality. And recently we did some modeling and have now adjusted those thresholds based on our CISNet networks modeling. And this is, I think, a tremendous population benefit. We've modeled the effect of more broad adoption of lung cancer screening nationally on lung cancer mortality. And we believe this is an underused approach that could be really, really important. Next slide, please. I'll say just one word about this, which is that there's a new technology for cancer detection, a blood-based assay and multi-cancer early detection tests. We are very interested in the possibility, the potential of these approaches. I think this is something that would be right in line with Sam's thinking about cancer early detection, and certainly possibly of interest to this new entity being discussed in Washington of ARPA-H, which might have special capabilities for cancer research. So stay tuned. I think a lot going on in this space. Next slide, please. And I'll close by saying that, in the 50th anniversary of the National Cancer Act, it's important to recognize the contributions of tireless researchers like Sam Gambier. And I'm reminded of this old saying that we drink from wells we did not dig, and we are warmed by fires we did not build. We make progress by building on these previous discoveries of great thinkers like Sam. I saw this quote in an obituary that was published in the reference below, and I really like this idea of, trying to prevent patients from ever coming to the hospital in the first place. Certainly a key goal of the National Cancer Institute and a legacy of Sam's work. So I'll close there, and thank you for the invitation for speaking today.
Thanks so much, Dr. Sharpless. It was great to see that the country has been fighting cancer for quite a long time now, and we're still not finished, but it's also encouraging to see that early detection and screening methods are really changing the way that patients are responding. With that, I'd like to introduce our next speaker, Dr. Ralph Weissleder. He's the Thrall Professor of Radiology at Harvard Medical School, the Director of the Center for Systems Biology at Massachusetts General Hospital, and an attending clinician in interventional radiology at MGH. The focus of his research lab is to obtain a deeper understanding of human biology and health and disease, to translate new biological understanding into clinically useful diagnostics, and to identify new therapeutic approaches and drug targets. He has published nearly 1,000 publications in peer-reviewed journals and has authored several textbooks. He is a member of the National Academy of Medicine, the American Academy of Arts and Sciences, and the National Academy of Inventors. We are delighted to have Dr. Weissleder joining us today to honor Dr. Gambhir's memory. Thank you for being here, Dr. Weissleder.
Thank you so much. You can see my slides?
Yes.
Thank you. Good afternoon to you. Thank you so much for the invitation to be here. It's really a pleasure to be in the session and talk about Sam's legacy and how we have interacted over the decades in an effort to improve cancer detection. So Sam and I go way back to the early 90s, where we were both interested in developing a new field, the field of molecular imaging. So the first 10 years or 20 years, we collaborated primarily in novel imaging biomarkers, then on gene therapy, then on nanotechnology. In 2000, so in the mid or early 2000s, we started to become involved in society work for the SMI, the WMIS, IS3R, the RSNA, and so on. And more recently, we sat on multiple boards together. During all of this time, we co-authored a book on molecular imaging and co-authored a number of publications, for example, this one here in Science Translational Medicine on imaging approaches to optimize molecular therapies. So as you know, Sam's background, at least the earlier background, was in nuclear imaging, whereas mine was more on the interventional diagnostic side, as well as the development of circulating biomarkers and liquid biopsies. And those two expertises were really complementary, and that is where we spend a lot of time in strategizing and developing new thoughts of how this might be brought to fruition in clinical medicine. So what I want to do today is talk a little bit about the circulating biomarkers and how they complement imaging approaches in our patients. And specifically, I want to focus on one type of test, and that is exosome analysis, or the analysis of other circulating extracellular vesicles. As you all know, exosomes are tiny little vesicles shed by cancer cells and a variety of other cells. So we have a fairly good understanding of how they work and what they actually do. So they're shed by cancers. They gain access to the blood, where they can serve as biomarkers, hence our interest. But we also know that they're shed into lymphatics, where they're taken up by subcapsular sinus macrophages, get ultimately overwhelmed, and then have effect of B cells promoting further tumor growth. Now, once they're in the blood, they have several characteristics which make them good biomarkers. They're fairly abundant, stable, they're accessible. They contain protein and RNA fragments of the parental cells, and thus reflect the cell of origin. So the common perception is, as shown here, that these vesicles have putative biomarkers, such as shown here, the CD63s of the world, CD81s, autotetraspanins. They contain multiple materials from the parental cell, and they have all of these functions. The reality, however, is somewhat different. First of all, there are multiple vesicle types, not only exosomes, but they're microvesicles and other particles with totally different ontogenies, completely different molecular biomarkers. They're produced not only by host cell, by cancer cells, but also host cell. There is no single defining molecular markers, and all of the markers are fairly heterogeneous and sparse. So with this, if we go a little bit deeper, an extracellular vesicle, an exosome, is about 1 million times smaller than compared to its parental cell. And this poses extraordinary problems in the analysis, the detection, the quantitation of biomarkers in these vesicles. However, they are abundant. But as you can imagine, early cancers that shed these vesicles in circulation are basically overwhelmed or overshadowed by the EVs from normal cells. And so in early cancers, the ratio of a tumor exosome to a normal exosome is 1 to 500,000 or 1 to 1 million. So in other words, if we process 100 microliters of plasma, there will only be about 10 events that we detect. And so you can see that this automatically becomes a signal to noise problem. And this is exactly where Sam and I had many, many discussions of how best to develop tests to crack this nut. At the end of the day, it also boils down to the fact that we really need to do single vesicle analysis, multiplex single vesicle analysis in large enough EV populations in order to identify those few vesicles that are shed by cancer cells. So my lab has spent a lot of time and effort in developing technologies. Many of them had their origins in nanotechnology to analyze these tumor-derived vesicles. More recently, technologies that allow one to analyze single vesicles, such as through imaging approaches, DEST approaches, PCR approaches, or what we call DD-seq approaches. So without going into details here, I wanted to sort of share some of the early results that came out of a project on studying intraductal papillary mucinous neoplasms, or IPMNs. And for those of you, for the imaging folks of you, IPMNs are becoming a bigger and bigger problem as these cystic lesions of the pancreas become more and more easily detectable with all this cross-sectional imaging that we're doing. It is currently estimated that at an age of about 60, about anywhere between 20 to 50% of the populations will have cystic lesions in the pancreas. Between 10 and 20% of these will ultimately develop into pancreatic cancer if left untreated. So they're being followed by annual MRIs, endoscopic ultrasound, and none of the imaging approaches is 100% predictive. So what is needed in this field are blood-based biomarkers to identify high-grade IPMNs or invasive IPMNs. So the key question, of course, is which biomarkers would predict a high-grade dysplasia or early cancer? And so we started by getting into this field by data mining through literature, looking at databases, atlases. We identified about 100 different molecular targets, tested hundreds of antibodies for these. And so this is what a typical list would look like. In the end, we decided to study every single biomarker on these exosomes that had ever been associated with pancreatic cancer, including those listed here. And from this, we derived what we call the MGH IPMN panel that we felt would be sort of in the sweet spot between something realistic and affordable and yet informative in terms of being able to detect invasiveness. Now, all of these markers here shown here had invariably been associated with pancreatic cancer. And so we were hoping that any of these would show up in exosomes of patients with high-grade dysplasia. So about 2 years ago, we started a prospective clinical trial where we enrolled patients, either healthy, age-matched, benign patients, patients with low-grade IPMNs, and patients with high-grade IPMNs, all of who underwent abdominal surgery and were followed by at MGH. And as you can see here, for the first marker, for MUC1, there was no significant difference between low-grade and high-grade IPMN. In other words, that exosome test didn't work. Much to our surprise, many of the other markers didn't pan out either. So what had been published in the literature, for example, TSP, DAS1, EGFR, GPC1, none of these showed any difference between high-grade dysplasia, early cancers, and low-grade IPMNs. However, there was a single one, and it was MUC5AC, where there was a clear difference between low-grade and high-grade. And so in subset analyses, we were able to show that exosomal MUC5AC is an invasiveness marker and is elevated in the earliest forms of pancreatic cancer arising within IPMNs. So if this biomarker is now added to either CT or MRI alone, the area under the curve for an RSC analysis goes from 0.6 to 0.99. So an extraordinary opportunity for increasing the specificity of our imaging tests by supplementing them with exosomal analysis. So as we think bigger and into the other forms of pancreatic cancer that are not associated with IPMNs, those arising from PANNs, the question is, can we expand this to the typical mutated proteins, KRAS, P53, and so on? And so one of the first questions we asked was, how common are mutated oncoproteins in EVs? And one could, of course, say, well, it's very simple. I just sequence everything. The problem with the sequencing approach is that exosomes don't have complete copies of the mRNA or even of the DNA. And it is estimated that currently there's only one copy of a P53, a mutated P53 per million EVs. And so that won't work well. So we hypothesized that proteins should be much more abundant. And we therefore analyzed and asked the question of how common are mutated KRAS proteins or mutated P53 protein in exosomes. So we derived EVs from cells that were all positive for either KRAS or P53 to analyze this. And at the end of the day, we were able to show that about between 1% and 2% of EVs or exosomes are KRAS positive. When imaged, the signal is usually sufficiently high to unequivocally demonstrate these mutated forms or oncoproteins in individual vesicles. So even in serum obtained from patients, such in this one here, where we imaged 1,600 vesicles, we were able to show that 1.24% of them were KRAS G12D positive, i.e. there is a malignancy in this patient. So with our current best thinking, one can take this, all of this data, and then derive mathematical models. And this is what Sam loved to do, to ask the question, well, with current detection sensitivities and the best diagnostic test out there, and with the existing knowledge of biomarker abundance and the antibodies that we have at our hands, what is really the detection threshold of what we're able to see in patients? And so this is sort of where we are right now. The blue curve on the right shows you the current detection threshold. And so we currently, for pancreatic cancer, so we currently can detect tumors as small as 0.1 cubic centimeters or 5 by 5 by 5 millimeters in 90%, over 90% of our patients. So our goal in the center here is to drive this curve to the left to ultimately be able to detect those millimeter by millimeter by millimeter pancreatic cancers in high-risk population, all through single EV analysis. I would also like to point out that the detection is not possible with bulk EV analysis because we would easily miss those rare EVs in the bulk analysis, as they would be overshadowed by signatures of the normal EV population. So I will stop here. I want to thank you for your interest and in commemorating Sam. Sam was a true giant, someone able to instill in all of us intellectual curiosity, apply mathematical rigor to what we're doing in the imaging sciences, and his ability to ask the right questions were really uncanny. So thank you very much to all of you.
Thank you, Dr. Weissleder. Great presentation. I think really captures the spirit of matching imaging with in vitro diagnostics, which was a big theme, of course, of Sam's group and many other groups. So moving on to the next speaker of this session comes from the private sector. And I think this, when my time with Sam, I watched him form multiple companies, and part of the spirit of his group and the molecular imaging program was really translating a lot of the findings from the lab to the bedside. When I left, the baseline study with a Google baseline study was just getting up and running. So our next presenter is Dr. David Suhy. David has a BS by way of background from the University of Pittsburgh, other training at Northwestern, and then postdoctoral training at Stanford. He's held multiple scientific leadership roles in the Bay Area in a number of companies, and is currently working on multiple therapeutic modalities in RNA interference, antisense RNA related to gene delivery. So please, David.
So the story of early is really sort of a story that Aruna sort of kicked off, which sort of described the way that Sam was very generous with his time. Early started because one of our co-founders, Cyriac Roding, who's now the CEO of Early, reached out to Sam, expressing his interest in Sam's research. And Sam, as kind as ever, wrote back to Cyriac very quickly. And although Cyriac was a serial tech entrepreneur up to that point, the two really became engaged in the possibilities of understanding and capitalizing on some of the research coming out of Sam's lab. And Sam was grateful enough to basically teach Cyriac biology at his kitchen table every other week for months on end before we physically started the company. And it really came out of Cyriac asking Sam, you know, of all the ideas that you've seen come out of your lab, and of all the ideas that you have seen propagated through science in terms of early cancer detection, which is amongst the most which is amongst the one that has the potential and the most promise. And Sam, and this became essentially the founding of Early, and I'll talk about it in a second. One of the remarkable things about Sam was his ability to connect with people on all levels. In fact, this picture on the far right shows one of the first company parties that we had. And it was actually, the company was so small at that point that I think the number of children outweighed the number of adults by almost 2 to 1 at this party. And I recall this party specifically because Sam, we had to schedule it through Sam's assistant because Sam was that busy. And it almost didn't occur because Sam was having a meeting with one of the world's foremost tech entrepreneurs who has started one of the world's largest healthcare foundations. And yet when we met for this party the very next day, there was Sam interacting with the young kids, talking to them about Einstein, pulling the "I'm going to steal your nose" trick, and basically having the children giggle as Uncle Sam went around this party. He's clearly a magnetic personality, and it clearly, to me, showed the depth of Sam's interactions and the ability to interact with world leaders of the foremost top levels, and yet the very next day be interacting with children. People have asked me, to describe Sam in a single sentence. I think we all sort of agree that that's a very difficult proposition to do. And the best way I can describe Sam in a single sentence is that he was an amazing clinician, an amazing scientist, but at the end of the day, he was an even better person. And I think that pretty much sums it up in understanding and really what Sam meant to us, meant to me in particular. So again, with this crowd, we don't need to go into this slide in depth other than to say that one of the biggest levers in terms of driving survival from cancer is the ability for early cancer detection. And really the concept of early came out of Sam's frustration to a large part of what was available to clinicians from an early cancer detection standpoint. So liquid biopsy, as Norman just talked about, is certainly becoming an ever-present and ever-increasing modality used for early cancer detection. As we all know, it's essentially taking a passively drawn blood sample from a patient and searching for naturally occurring biomarkers using chemistry, all the while hoping that any biomarkers are present in the serum whatsoever. And certainly, while it's a technique which has great validation and great clinical promise, in fact, with stage 3, stage 4 cancers, the numbers have been really great. For earlier cancer detection, where you might be talking about stage 2 or even stage 1, across a great number of cancer types that have been handpicked by companies like Grail, Freenome, Thrive, the numbers drop off dramatically. And that only makes sense because essentially the amount of biomass available to shed those biomarkers naturally into the bloodstream becomes less and less. So let's say that even if they got it to 100% and the search does deliver a signal in the blood, the tumor still may be too small to image. And we've heard certainly a lot of localization talks, and certainly it doesn't show which lesion is malignant. Now, what are you going to do except for worry about having this cancer? It's going to be too small to essentially treat with chemotherapy, and you can't cut it out with surgery because you can't locate it. So along what Anna Wu suggested, Sam had the big what if questions. And the big what if question here is, what if the early cancer became obvious? So obvious that it's like shining a bright light to itself. So what if, and this is Sam's big what if question in regards to this particular technology, what if you could get an injection that releases a DNA vector that enters healthy cells, and if there's any present, also cancer cells. But if, and only if it's a cancer cell, will it force the cancer to produce a non-human synthetic biomarker such as limonene. Limonene is in oranges and lemons, not in humans unless you've eaten them. Yet, if you've taken a vector that forces your cells to express limonene synthetase, and you breathe out limonene, you may have cancer because the cancer was forced to produce it. Now, at that point, you're really not better than some of the liquid biopsy companies because you detected a problem, but not a specific problem. So now if the test is positive, you can also take that same construct, genetic construct, to physically produce an enzyme that digests a radioactive molecule, a PET reporter gene. Now you know perhaps the exact location of the cancer in the PET scan, and certainly we've heard a lot of talks about this today. And then finally, you might be able to use precision radiation to focus on that specific localization of that cancer for precision killing, or even to have a surgeon cut it out. And in the future, you might even think about using the same platform to trigger a kill cascade. So how it works, it's essentially a DNA reporter, but it's much more than that. So in this DNA reporter, you've got a non-viral nucleic acid template in which you've written an if-then clause. If cancer, then produce something. The if takes advantage and usurps the very dysregulated pathways that cause the malignancy in the first place. And John Ronald in Sam's lab showed this initially using the survivin promoter, which is highly regulated, upregulated in a number of cancers, although not all cancers. Because it's a genetic system, we have the ability to multiplex and write multiple and/or logic gates to increase sensitivity and specificity. In addition to that, you have the ability to use chimeric promoters and regulated sequences to create high degrees of specificity. We worry as much about making sure that the promoter is shut down within the context of normal cells as it is upregulated within the context of cancer. And likewise, we want to find things that are not highly upregulated in comorbidities. The reporters we've talked about, limonene might be a breath-based test, a PET imaging marker, or in this case, what we'll talk about today, it might be secreted embryonic alkaline phosphatase for a blood-based assay. And then finally, there's a delivery agent. Now, the delivery agent's responsible for making sure that the molecular construct not only gets into the cancer, but also into the nucleus for efficient expression. And you might say, well, that's great, David, but gene therapy companies have been around for the last 25 or 30 years. But again, here's where we have a paradigm shift. Within the context of a therapeutic, you're worried about transfecting as much of the disease tissue as possible and avoiding off-tissue targeting. So you're talking about adding tremendous amounts of nucleic acids where you might look for 95, 98, 100% transfection efficiency of a specific tissue. Within the context of a diagnostic, you might want broad distribution properties to sample many different tissues. You also don't need necessarily 95, 100% transfection efficiency. You might not need 50, 20, 10, or even 5%. Simply, you need to transfect enough cells and produce enough of a signal within an appropriate signal to noise ratio versus normal tissues for detection purposes. This dramatically lowers the burden in terms of utilizing these constructs. And quite frankly, from somebody who's been in the gene therapy field for the last 25 years, this in itself is a complete paradigm shift from traditional use of nucleic acids for detection modalities versus gene therapy agents. Now, John Ronald at Stanford showed this very elegantly in using DNA minicircles, went into xenograft containing mice, injected into tail veins, and compared basically the output, which in this case was secreted embryonic alkaline phosphatase in tumor-bearing versus non-tumor-bearing animals. And at Stanford, John and his colleagues with Sam published this data back in 2015. And what they were able to show is the production of secreted embryonic alkaline phosphatase in tumor-bearing mice shown in the red versus non-tumor-bearing mice shown in the blue. And they saw a 3.7-fold change in terms of the amount of secreted embryonic alkaline phosphatase. So when we started early, we took upon ourselves the idea to bioengineer this problem, focus on multiple aspects, including delivery agents, types of promoters, types of reporters. And at the end of the day, using these bioengineering approaches, we exchanged DNA minicircles to an alternate DNA template. We iterated on compound concentration, firmed up delivery formulations. And what we're able to show is the ability to increase the signal to noise ratio dramatically within the context of essentially the same tumor bearing model. Now, to illustrate the concept of distribution, what we showed is by pulling apart this animal and looking at the vector within the various different types of tissues, you see you have the ability to sample a large number of different types of tissues. With this particular delivery agent, most of it went to the lung, but certainly there was measurable copy numbers in other areas. Now, this is a transient-based effect, and these plasmids, or these DNA templates, are meant to be episomal in nature. And certainly what you see is that over time, biodistribution basically cuts out and the DNA gets dissolved naturally over time. So this is a transient-based effect and non-integrating in nature as a whole. Now, we tested this in a large variety of different types of models, including a metastatic lung model, where you see an incredible signal to noise ratio associated with high tumor burden, syngeneic lung models, as well as altering up the delivery compound to make sure that this wasn't specific necessarily for lung tumors. But we used, in this particular case, in the last panel, a lipid nanoparticle to be able to deliver and look at signal to noise ratio in orthotopic liver tumors. Now, the question then becomes is how small can you get in terms of tumor burden within these animals? So we've used a bioluminescence-based cell line and showed in this metastatic model that in the first week of tumor uptake, the majority of these cells localize to lungs. That's what they're designed to do. This is a small number of cells, the equivalent of a 1.4 millimeter cube tumor. And what we show is that the lung weight barely changes at the week 1 when you compare the light pink bar to the gray bar. But even at week 1, you're seeing a 10x signal to noise ratio. By week 3, 27x. And by week 4, where the line metastasizes and the tumor burden becomes more spread throughout the animal, we're seeing now 129-fold signal to noise ratio. So what we're doing to improve this, we've talked about the ability to improve the types of templates, the different types of sequences that we can write and/or logic gates. But at the end of the day, this is nucleic acids, and delivery is a key feature. And so what we've been working on is utilizing novel tools by applying some new technologies to be able to better develop delivery agents specifically, which will be more targetable to cancer cells in vivo. One of the ways we've done this now is to start transfecting animals and pull the animal apart and pull the tissues apart bearing the lungs, and now start separating those tissues into independent single cell suspensions using molecular markers to trace lineages of the proteins, as well as 10x single cell genomics to be able to ascertain where the cells were derived from. Specifically, why we're interested in doing this is that most cancers formed inside the body are epithelial of origin. And so if we're going to really take this to the next level, we need to start understanding not only where our vectors wind up in terms of tissues, but also making modification to these delivery agents, which proportionately skew the ability to transfect epithelial cells within an in vivo system. And this is some of the data that you see here. And we're not disclosing essentially where these are, but you can see that very specific cell populations as identified by this 10x technology can now be transfected. So now that everything that I've showed you has been in a murine model, and certainly murine models are fine, and they recapitulate some aspects of tumor biology, but we'd like to be able to scale the technology on the way to human beings. Dog is one such animal model, and if we had the ability to go from a mouse to a dog, it would be substantially larger than going from a dog into a human being. In addition to this, dogs develop cancer much the way human beings do, spontaneous growth of the tumors. So there's no artificial vasculature in terms of setting up these animal models. What we've done is now, the problem is that there's no really great laboratory model of canine cancer, but what there is nearly 6 million dogs a year annually in the United States diagnosed with canine cancer. And pet owners that bring their dogs to the clinic for treatment. So we've set up collaborations, one with Michael Kent, which is the data I will show you here up at UC Davis, where we have the ability now to test drive this technology within the context of spontaneous canine cancers. We set this up as a traditional 3+3 and moved forward and looked at the ability to produce a cancer activated signal within these dogs. You can see at the low dose of this compound, which we call early 001, you can see that the gray dotted line represents normal dogs, dogs without tumors, which had received this dose. Lower limit of quantification of the assay is shown in the dotted red line. So any signal below that is essentially not a cancer activated signal, and it's not measurable as well. Even with the low dose, we see one out of 3 dogs has somewhat of a signal that's being produced. In this case, it is secreted embryonic alkaline phosphatase. We know that the human promoter that we're using in this construct works just as well in dogs based on sequence homology and testing in cell culture lines. Now at the mid dose, you clearly see in a wide variety of different types of cancers, this transient signal, which comes up by day 3, day 4, and essentially progresses back down to baseline levels. And certainly at the high dose, we see a universal response once again, even though we're working with some tumors where we think the bioavailability of the compounds is quite low, such as oral melanomas. We've been able to isolate some of the tumors out of a portion of these dogs and show that there's been a responsiveness, for instance, of Ki67 as a general proliferative marker, and show that we have the ability to detect the early 001 construct in several of these tumors, albeit it is a challenge to be able to get some of the surgery performed in a timely manner that allows us to really maximize the amount of DNA copies that we can detect on a per cell level. And then finally, we show in this case, this was using the survivin or BRC5 gene, that we know that there is high endogenous level or higher endogenous levels of the BRC5 transcript as compared to the normal dogs. So the last slide I'll show here is that this has really pushed us forward. The company has initiated a first-in-man phase 1 human clinical trial in Australia. We're initially working on late-stage lung cancer patients, and this will be with a molecular construct that uses the secreted embryonic alkaline phosphatase. We're performing this trial at 2 of the 3 largest clinics in Australia, and the readout will be the amount of secreted embryonic alkaline phosphatase versus traditional PET-CT and metabolic PET to assume the relative tumor burden. And it's our hope we have the ability to correlate the 2 values. So the last slide is just my colleagues at Early, thanking all of them, as well as contributions from the Stanford lab, in particular to John Ronald, whose initial input into founding the company was important for us to develop some of these technologies. So thanks very much. Appreciate your attention.
Thank you so much, David. Very exciting to see some of this work moving so far along, even into people. So moving on, I think, to our last speaker of the session. Before we do, I just want to remind everyone that you are able to put questions into the chat box on the right side of the live stream that we will address when we move to the discussion section of this session. The last speaker of today, I'm very excited about because the word that comes to my mind with Dr. Sangeeta Bhatia is creativity. And I've been reading her papers since I was a PhD student. And creativity is one of the things that always really impressed me of Sam. And so it's really exciting to have her here today. I'll do just a brief introduction for time. And she is currently at Massachusetts Institute of Technology, where she holds many awards. But I do want to mention HHMI, National Academy of Science, National Academy of Medicine, and National Academy of Engineering. So please, Dr. Bhatia.
Thank you so much. Can everyone hear me okay? Just a thumbs up.
Yeah, looks good. Sounds good.
I am, it's a real honor to be here to remember and honor Sam's legacy today. I thought I knew a lot about him, and I'd read many of his papers and met with him many times, but even just experiencing the breadth of his work and impact today has left a mark on me. So thank you all for putting it together. I'm going to tell you about our work together in synthetic biomarkers. which you just heard about from David, which is the idea that you could make the body make signals that are not normally being made in the hopes of finding cancer earlier. And my sort of journey with Sam starts actually when we were both in Southern California about 20 years ago. So that's me at UCSD and him at UCLA. And I only knew about Sam back then as this kind of rock star that was doing molecular imaging. And I only knew him by reputation. And it would take the CCNEs that you heard about, the Cancer Centers for Nanotechnology Excellence, for us to meet. By then I was at MIT, and he was, of course, at Stanford. And I think we've heard a lot about Sam's impact on his trainees and how people found him and how much time he spent with them. And I felt really lucky to sort of have been collected by Sam or discovered by Sam at that point as an intellectual colleague. And he subsequently invited me to speak at the Canary in 2009. And after that, I think that, we had many conversations and I just sort of came on his radar and he kind of pulled me into his world. And the next thing that happened was that he asked me to sit on a study section for this organization in Houston, the CPTAC Cancer Prevention and Research Institute, where he was a study section chair, and he had pulled together a lot of his colleagues and former trainees to evaluate grants. And there I got really to see another side of Sam. And I got to see his big brain, which I knew about, but I could just witness his comprehensive knowledge. And how people have talked about how funny he was and his wry sense of humor, but that would come across as he was sort of getting us through our study section day. And he was a great listener. He would take everyone's opinion, but he had a very strong and specific view, and I think that's very characteristic of him in sort of his being a visionary and asking us all to think bigger about what really matters. And the other thing that I learned about him in our time together in these study sections was about his big heart. So not just the big brain, but the big heart. And people have talked today about how he was such a good human being. But I got to see that he really lived his values. If you just looked at the composition of that study section that I was on, he had his former trainees, and actually quite a number of women. People haven't talked today much about how he really was very inclusive, but that was where I first met Anna Wu and Kathy Ferrara, and there were a lot of amazing women in that room, and I really appreciated. This is sort of, before it was in the cultural zeitgeist, I think, to be as inclusive as he just naturally was. So I really appreciated that. And then we would serve together at the NCI, and I would see how he would advocate for the field as a leader. And then I would start to learn about his views on commercialization and his experience in regulatory and, this quantitative analysis that people have talked about, and how he would really think about how much money it would take to get these early stage technologies in early cancer detection out into the world. And I learned so much from him about how to think about that problem. Then in 2011, he published a paper with Sharon Horry, which I'll show you in a second, which was really my personal inspiration for coming into this field of blood biomarkers and possible ways to do better than blood biomarkers. And it was a really classic Sam paper, and I'll show you a snippet in a second, where he used really mathematics to answer a question about how could we do better. And then he would start to come to Boston, for he was working with some VCs, and I got to see more and more of that sort of side of his brain and how to think about startups. And then I started a center at MIT called the Marble Center for Nanomedicine, and asked him to be an advisor. I knew he was so busy, but we just had to have his insight, and he accepted, and we were so lucky to have him give a plenary at MIT in 2018. You saw the snippet of it earlier in the compilation, where he visited us and shared his vision. And then together we worked with this international consortium on early detection, which was stood up by the CRUK. And that was actually the second to last project that I worked with him on, and he was so passionate about it. And we we together were in Portland, Oregon at OHSU around 2019. And this was the first time that I heard him talk about Millen and Millen's journey and how it could have been different. And it so touched me because it was so typical of Sam, because it wasn't just his work. He would pull in, every dimension of his experience and think about how can we have a different reality. And then finally, we were asked together by the NCI to run a think tank together on this idea of synthetic biomarkers. And by now, our two labs, we're working in this space. And Sam said to me, some people would think that we are competing. But of course, we're not competing. We are both focused on the enemy of human disease. And let's have this think tank and let's just pull everybody into the field who's like never thought about how to make your own biomarker. And so we designed that meeting together, and you can see a snapshot of it here. He actually couldn't come in person, so it was in July of 2019, but he gave the plenary, he taped it ahead of time, and we have a think tank article that's just been accepted to share the conversation with the community. And you can see there's all kinds of folks in the room, including some of his former trainees and clinicians and imaging folks. So it's just very characteristic of Sam that to try and kind of open up a field with a colleague. And then this map is just to show you the places I physically interacted with him. And I know that he was really all over the US at every moment. I just got glimpses of him over the course of a career, and he was such an inspiration and continues to be. So just to tell you a little bit about the synthetic biomarker work that we've done together to build on David's comments. I think it's well appreciated, and as you've heard, that if we could find cancer earlier, it would be more treatable, and there's lots of great data about that. This is this one snapshot, and of course we can use imaging to do that, but imaging has limitations. And of course, we can use blood biomarkers to do that, but they too have limitations. I think people knew this and we thought a lot about it, but this paper that I described from Sam is the one that really inspired me, Sam and Sharon both, where he just did a thought experiment, which how good could you ever get if a cell made as much protein as it could, as fast as it could, and it diffused into the bloodstream, and you had a certain amount of healthy cells that were making a background, and then that protein product had a certain half-life in the diluted bloodstream. Like, how good could you ever get? And he asked the question together with Sharon in the context of ovarian cancer, and found a really disheartening number, which was 10 years after initial tumor initiation. And then he did a very same thing, which was to ask in the paper the questions, what ifs? And the what ifs in the paper are every one of these. What if we used a different instrument? What if we had, and so on and so forth. And you could see like how much better you can get. So this paper really inspired our thinking, myself and my trainee Gabe Kwong, as we went into this field and tried to think about how to overcome the limitations that they outlined in this paper. The background secretion by healthy cells, the dilution and degradation of analytes in the blood, and sort of the analytical difficulties of quantifying things in the blood. And so we were trying to think together how to go from endogenous blood biomarkers that are diluted and degraded with fixed analytical systems to a new paradigm. And the sort of version of synthetic biomarkers that we've been putting forward is not so much in the imaging or secreted enzyme space, but in this space where we give sensors. So we give exogenous probes, and they're decorated with peptides. And when these probes encounter tumor-associated enzyme activity, like proteases, those proteases can cleave a reporter analyte. And the reporter analyte is designed to allow signal amplification. So one copy of an enzyme can liberate many of these reporters. The reporter analytes are synthetic biomarkers, so they're not present in the healthy human. And they're designed chemically to resist degradation. So we use a trick of making them out of D amino acids. We know that they'll be liberated into the blood, but we can also take advantage of the fact that the kidney is a natural concentrator to get them into the urine. So that's a 20x concentration. And then they find their way into the urine, and in a healthy patient, there's low background in the urine. And then because these are completely synthetic systems, you can really design the reporter analyte to be compatible tool with the analytical tool of choice. So you could use something like mass spec to be highly sensitive. You could use a clinical ELISA that would be easy to deploy. I'll show you in a moment that you could put it on paper for the point of care. And then importantly, and sort of reflecting Ralph's talk, multiplexing is really important to both sensitivity and specificity. And so ideally you'd be able to do this with multiple probes and then multiplex the signal for predictive accuracy. So this is the idea that we've been working on, and we've chosen proteases as a class of enzymes to trigger this reaction, although one could use others. They have lots of important advantages for this, but the most important amongst them is their well-known involvement in cancer progression for decades. Most notably, and the example I'll show you will build on this one, MMP9, which is a type 4 collagenase, which is important as cancer invades the basement membrane and spreads, and also comes on in the angiogenic switch. So when cancer outstrips its blood supply and has to recruit blood vessels about 1 millimeter in size. So it's sort of a nice candidate from which to start. So that was what we chose. This is just a little video that we made to sort of describe this process. I think this is something that Sam did wonderfully, is visualize his sort of dream. So here the dream is that these nanosensors would be injected into the bloodstream. This tumor is surrounding the bloodstream, has triggered the angiogenic switch, so the vessel is hyperpermeable. And these nanomaterials can then extravasate into the interstitial space of that tumor. They've been designed to be activated by that MMP9, so the type 4 collagenase, which are these yellow balls in this picture. And each one of them can then activate a sensor and liberate many, many reporter analytes. And these reporter analytes now are designed to be stable, as I said, in the circulation. So they're made of D amino acids and can no longer be degraded. And when they find their way now back into the bloodstream, they can take advantage of renal concentration because we know that the kidney is essentially a 5 nanometer filter. So they're smaller than 5 nanometers, and then they concentrated into the urine. And this whole process happens over the course of 1 to 2 hours in mice, in large animals, and also in humans. So this is an idea that we put forth in a paper in 2013 that was inspired, as I said, by Sam's 2011 paper, and something that we've been working to advance in a series of papers and a startup over the years. And the data I'll show you today is looking also at lung cancer. And in this example, what we've done is made a cocktail of these materials, so not just one sensor at a time, but a 14-plex. So each one of these is designed to be cut with a different sequence, and it has a different reporter. And the reporters in this case are bar-coded by mass. So you deliver them here into the pulmonary space, so they query the lung preferentially. The reporters are liberated, as I said, after an hour in the urine, and then you mass spec the urine for the barcode of what happened to those 14 different materials. So we've quantified, sort of in the name of Sam, the sensitivity of this system, and it is quite sensitive. In mice, it's more sensitive than circulating DNA using current methods. The limit of detection is about 3 cubic millimeters. It's quite specific. We've gotten up to 19 probes at once. In typical mouse model, so mice are isogenic, of course, and it will be more complicated in humans, but in mice we find that it takes about 3 to 5 probes in any one cassette to classify disease quite robustly. And we've worked, because this is a completely synthetic system, on many, many different versions of these formulations. A sustained release one, an injectable one, as I'm showing you here, a powdered inhaled one, and we've worked on different readouts. And so in this paper, which we published in 2020, in the lung cancer example, we looked in 2 different mouse models of lung cancer and compared the ability to call cancer in the earliest stages, so 5.5 weeks of tumor initiation, to not just benign normal mice, but also benign inflammation. and showed a really strong classifier, again, just in animals. And then actually we published a paper in 2015, which was an echo to Sam's 2011 paper, which asked all the same questions that he asked in that initial paper about the natural blood biomarkers. We asked it with our system, which was how good could this be? How many enzymes are present in the tissue? How many reporters could you liberate? And so on and so forth, and did that sort of allometric scaling to humans. And so we continue to do that as we improve our system using kind of a roadmap that he laid out for us. So we've used this in about a dozen diseases in animals: cancer, fibrosis, which is proteolytic, thrombosis, and infectious disease. And we have a startup that has last year completed a phase 1 clinical trial. So is poised now to sort of do the pivotal work. So we're really grateful to him for all his thought partnership over the years. I'll just close by saying Sam has also inspired us to think beyond detection, to think about monitoring treatment response, and then also surveilling for recurrence. And so in this example, what we did was take those EML4 ALK mice and treat them with a small molecule and show that one of the sensors is really sensitive to tumor regression. And that we could then use this to classify treatment response by doing this longitudinal urine test. And the very last thing that we're starting to do, this will be my last slide, this paper actually came out last week and we dedicated it to Sam, was load these materials with copper-64. And the idea here is that you could monitor patients with the urine diagnostic, and then if you had a cancer call, you could load that material with copper-64 and then do a PET image to identify the localization of the tumor. So I really want to thank you for your time and attention, and just say I've been part of a number of pieces of work in the last year that have given dedications to Sam, and you'll see them here. Maybe I'll just read this one, which is by the Early Detection Consortium. We dedicate this to our friend, colleague, and mentor, Sam Gambier. He chose us to early detection of cancer and taught us always to look to the horizon, which is, I think, so apropos. He really did that. Actually, I'm co-chairing a conference in January on early cancer and prevention, and we'll be dedicating that to him too. So thank you so much for the honor of being here and remembering Sam together.
Thank you so much, Dr. Bhatia. Really, really inspiring work. Again, feel free to put any questions you may have into the chat. As those come in, I might start off with a what if question. What if one of these works with 95% accuracy, affordable, but we can't get people to use it? So I'm thinking along the lines of vaccines and vaccine hesitancy. What are the tools that we use to make these detection schemes more used by the general public? Maybe Dr. Sharpless.
I mean, we kind of know the answer to this because we have an effective lung cancer screening test now that is widely underused. 5% uptake would be optimistic. It clearly benefits these patients and can reduce mortality. We also have, preventative vaccines like HPV and Hep B that are widely underused, although, maybe slightly better statistics there. The NCI has a long experience with this issue. In fact, we were one of the groups in the federal government that was tasked to write a vaccine hesitancy kind of report around COVID. And so many of the people at NCI have been working on HPV vaccination, for example, help contribute to that effort that has now been widely disseminated by the CDC and other parts of the federal government. But I think, the answer really appears to be for screening and prevention strategies to get to the doctors that care for the patients. That is perhaps more important than any other, combined with education and payers and a lot of issues that are complex. Basically, patients listen to their doctors. And so that you want the family practitioners, the internists, whoever's going to be seeing these patients when the patient is appropriate, to hear from their physician that this is something they should do.
David, any perspective from the private sector?
Yeah, I think Dr. Sharpless hit the nail on the head. I think that if you can get practitioners to buy into the use of some of these early technologies, it would make a significant amount of sense. I would argue that trying to attack this through the patient perspective, it's interesting. Certainly we've seen this with some of the other early cancer detection modalities where we've all seen the commercials on the Super Bowl for Cologuard or other sorts of implementations of some of these technologies. But at the end of the day, it's still going to be the medical practitioner that will likely drive the majority usage of these tests moving forward.
Sure.
Thank you. We have a question online for Dr. Weissleder. Someone anonymous is asking if they wonder if the 1.24% EV KRAS positive is enough to determine pancreatic cancer. And does that mean that the control blood has no extracellular vascular KRAS?
So of course there is background. There's background because KRAS occurs, or KRAS mutations occur in the normal body as well, number one. And number two is because we're using antibodies, and so they will have some background. And so it's a question of defining what that background is and establishing criteria to be above that background. So it's the same as in imaging. You have to have a signal to noise ratio above that background. And with that background established, and it is not zero, we're currently able to detect cancers as small as I said, so the 3 by 3 by 3 millimeters in size.
Thank you.
And a question for Dr. Bhatia. How would you make sure that the synthetic biomarkers are less than 5 nanometers for effective kidney filtration? And can you control the size? Proteases are probably pretty well defined.
Yeah, that's a good question. So the way the reporters are designed is that uncleavable part, the D-amino acid part, is a certain number of amino acids, and that just keeps them under that filtration level.
Great.
I had a question for Dr. Sharpless about sort of how it was really interesting knowing that it's the 50th anniversary of the Cancer Act, which I had no idea about, and that was a little before my time, but it was really interesting to see that it's been 50 years and that they were projecting a cure in around 3 to 4 years. And I thought that was interesting. Cure, the idea of curing cancer. Can you speak to what NCI, how they defined cure? Because cure means a lot of different things. Maybe it means like, oh, we'll never even get cancer, but maybe cure means something else now. It has a different definition.
Yeah, I think, the relevant historical context there was, antibiotics had transformed infectious disease from, everybody died of sepsis at one point to nobody dying of sepsis practically. That I think was, the legislators had that in mind, and many of the principals involved, spoke in these terms. And then also there was a remarkable event in the sort of 1950s and early 1960s where pediatric leukemia, ALL in particular, went from a death sentence to sort of a 90+% cure rate. And so, if we could do it for pediatric ALL, we just have to do it for like everything else, was the thinking. So, infectious disease plus the paradigm of a single disease having, and there were a few others, choriocarcinoma. There were a couple of things where there had been some progress. So I think that was the expectation was, this will be easy. And I don't know that they defined it so much as cures, just like cancer wouldn't be a problem. We either have effective prevention or effective early diagnosis or effective treatments or some combination thereof. And, I think that really, it shows that at that time, the basic biology of malignant disease was sort of an unknown unknown. I mean, we really didn't even know what we didn't know back then. And that now it's really changed. I mean, you can see, we know how cancer works for the most part. We just don't necessarily, we know we need to drug RAS. We just don't have like the right way to do it, but we think that's a tractable problem. And that is a much better problem to have than like not even knowing where to start. So I think that there has been significant progress, but it's still a ways to go. We at the NCI have embraced the C word. You know, we backed away from it in medical oncology for many years because I think it was very confusing to patients and caused some problems. But you know, whether or not we in the medical oncology community are comfortable with that term, the American public wants that, and the legislators that fund the NCI want that. And so, you know, I think we have in the last few years, particularly since I got back to the NCI, have been talking about curing patients, and that's our goal. And we do that in like metastatic melanoma now. That is a cancer that we make go away and never come back in the majority of patients. And that used to be the worst cancer imaginable when I was a fellow in the metastatic setting. And now it's largely a curable entity. And we have similar examples in other solid tumors like lung cancer, et cetera. So, you know, we are curing patients, but not enough. And certainly, I don't mean to imply that therapeutics is the whole answer. I think prevention and early screening and, you know, survivorship of chronic disease is actually, you know, we have people living with breast cancer decades now, and that may be a good solution in certain patients. So, you know, we just want to end cancer suffering is our current goal.
Great. There's a couple more technical questions for Dr. Weissleder. One from Jan Grimm about reaching a threshold where you're detecting mutations that are not yet clinically relevant. And this kind of maybe goes back to the previous question about background, but could you comment maybe on detecting these ongoing mutations that are weeded out naturally by the immune system?
Yeah, so there is not a whole lot of data out there. So we know that we can detect them. And so our ultimate goal is to improve the specificity of a test by actually looking for those ultra rare EVs that are double mutated, because they unequivocally have to come from cancers. And so the race is on for multiplex detection in single EVs. I mean, that's all I can say at this point.
Sure. And then another question from Melissa Burkett about the method of isolating extracellular vesicles. Of course, a very difficult problem. She's mentioning filtration systems along the line of a dialysis machine.
Different isolation methods. And at the end of the day, we gave up on all of them because they were either too lengthy, too costly, or they could not be translated into a clinical workflow where you have to process dozens or hundreds of samples in a hospital. And so what we do right now is we're looking at every single vesicle in plasma, and we're basically using glorified Amicon filtration discs to get to that. And we analyze hundreds of thousands of vesicles, and that's basically what we're looking at. So we're not doing ultra pure purification of one specific vesicle type and losing a lot of these vesicles in the process of purifying them, but we look at everything at the single vesicle level, agnostic of where they came from. And so that's a much easier problem to solve.
It's spinning your wheels to get a
Right,
very
exactly.
purified
Yes.
subtype.
Exactly.
Yeah, yeah, great. Okay, well, I think Cristina, do we have?
There's just people saying what a wonderful talk each of you gave. That's definitely coming through in the comments. They're all very gracious for your words.
One last question for Dr. Suhy. Could you comment maybe on plans for trials in North America? I know you mentioned you're primarily working right now in Australia.
Yeah, I can. So the idea is that the Australia ecosystem for us is a little bit quicker, although we have had discussions with the FDA in terms of being able to run our later stage trials in the United States. FDA certainly understands the program, but for us, since regulatory is run more at the state level and at the institutional level within Australia, for us, it became an ecosystem for us to get into the clinic with a little bit more expediency. Certainly though, notwithstanding COVID lockdown, which is far stricter in Australia right now, it has been a little bit more challenging to enroll patients. But once the restrictions lifted, we anticipate running through that trial very quickly and then importing the data back into the United States for the later stage clinical studies, phase 2, phase 3 studies.
Great, great. Well, I think we are at the, we've used our appointed time. On behalf of all the organizers, I want to thank everybody in this session for making the time to commemorate Sam, and we'll be back after a 10-minute break.